Greetings, this is Dr. Mubin from the Department of Zoology. Girls, last class we had dealt with what is called as cell cycle, right? So today is a follow-up of cell cycle. What is the part of cell cycle where the division takes place? This is a cell cycle, right? What we did last week. So within the cell cycle you have the G1 phase, synthesis phase, G2 phase, followed by the M phase. Today we are going to deal with the stages of the M phase. What is this M phase? Which is called as the mitosis phase, which is the cell division phase. This mitosis takes place in what is called as the normal body cells. So let us see what are the stages involved in M phase, which is the prophase, prometaphase, metaphase, anaphase and telophase. Now let us see each and every stage in detail. What are the events occurring in every phase? Let us start with prophase. Before prophase, we have what is called as the resting stage, which is the interphase, where actually the division takes place. The G1 phase, G2 phase, S phase, all three together constitute what is called as the interphase. Now after interphase, during this interphase, what happens? Cell replicates its nuclear material, ensuring that when it does not divide at the end of the mitotic phase, each of the newly created daughter cells will contain the full set of gene. So it's just a preparatory phase before the cell division phase. Next. When you talk about prophase, this is the first stage of mitosis. So during mitosis, in the first stage, the first nucleolus phase, you can see the nucleolus inside the nucleus. Nucleolus inside the nucleus phase or disintegrates basically. Chromatin, which is the replicated DNA, condenses into what is called as chromosome. In the interphase stage, we see, right, the chromosome is like a thread-like structure. But whereas in prophase, the chromatin material condenses to form what is called as the chromosomes. You can see the four arms of the chromosomes very clearly. So they condense to form chromosomes. And each replicated chromosome comprises of two chromatids, both with the same genetic information. So this is the first stage, which is the prophase, where the nucleolus starts to disappear. The nuclear membrane also slowly starts to disintegrate, and the centrioles divide and start moving towards the corners, so that they get ready with the formation of the spindle fiber for the second stage. So three basic major events take place in the prophase. Nucleolus disintegrates, nuclear membrane tries to disintegrate, centriole divides into two and tries to move towards the corner. Next stage. This is how the prophase stage under the microscope looks like in an animal cell and a plant cell. The difference between how a prophase photomicrograph of a prophase stage looks under the microscope. Next. The second stage is called as the metaphase. As the word says meta, half. So we can see the arrangement of the chromosomes in the central plate, equatorial plate. Okay, so here the nuclear envelope breaks completely and a large protein network called the spindle attaches to the chromatids. You can see here these yellow color lines. These are called as the spindle fibers, which are starting. This is a protein network basically. So from the centriole, these spindle fibers run down from both the sides and attach to the chromatids. And this red color dotted line, it is called as the metaphase plate, which is in the center, and to the center region the chromatids are arranged, the chromosomes are arranged and they are attached to the spindle fibers. So the chromosomes are now aligned perpendicular to the spindle in the equatorial plate. They are not parallel, they are perpendicular. Only then the centriole can divide into half and two chromatids can separate in the next phase. So this is very, very important. The chromosomes here attach to the metaphase plate in the perpendicular plane. Is it clear? Next. This is how a metaphase stage looks in an animal cell. You can see here, this is the thick, darkly stained chromosomes and these are the spindle fibers. This you come across in an animal cell, and this is how the plant cell metaphase stage looks like. You can see here, this pink color structure with light blue color spindle fibers. This is again the photomicrograph. I am showing you comparison of both animal cell and plant cell for every stage. Next. Then comes what is called as the anaphase stage. Now this is the stage where actually the chromosomes which are attached at the centromere, they try to spread apart. You can see here, it was actually two arms, right? So here the centromere breaks down because Now an independent chromosome with own centromere begin moving towards the opposite poles. So the movement is made possible via two mechanisms. One, the kinetochore fibers attached to each centromere begin to shorten. They reduce in size. They begin to shorten and pull them across the pole. At the same time, the polar microtubules, that is the part of the centriole, the polar microtubules from opposite ends of the cell form cross bridges in the equatorial plane, pushing the poles apart for becoming longer so that they can completely move and separate. Next. So this is how a photomicrograph of an anaphase stage looks like. You can see here two clusters of chromosome just being pulled apart to two poles or two corners. This is in the animal cell and this is in the plant cell. You can see cluster of or a group of chromosomes being pulled to either side. Next. Telophase and cytokinesis. This is the last phase. See, when we talk about division of mitosis, it's basically two parts, karyokinesis and cytokinesis. Division of nucleus is called as karyokinesis. So up to telophase stage, it is division of the nucleus. After which we have what is called as the cytokinesis, which is the division of the cytoplasm. So what happens in telophase? Let us see first. So in telophase, the daughter chromosomes, you can see now the violet chromosomes, arrive at the poles, at the spindle fibers. You can see the two poles they have come. So after completion of separation, the chromosome, the nuclear membrane begins to reform, rearrangement, because it's only disintegrated. It is not dissolved or gone from it. It is only disintegrated and present inside the cell itself. So once this division is complete, the nuclear membrane tries to rearrange themselves. So the nuclear membrane starts forming, the nucleolus also starts forming, and the spindle fibers start disappearing. Right? The spindle fibers start disappearing. So what happens is within the same cell, you will have two nucleus. You will have, you can say, two nuclei. So within the same cell, you will find two nuclei, which is a small invagination in the center. So this phase is called as the telophase. Okay, so when the telophase is complete, the new cell membrane is being formed. So the final step of telophase involves the initiation of plasma membrane cleavage. You can see this, the cleavage furrow here. So they start invaginating inside. Initiation of plasma membrane cleavage between each of the new daughter cell to ultimately yield what is called as two separate cells. Is it clear? So with this we come to the end of cytokinesis. Okay, so this is how the telophase of animal cell look like. You can see here you have two cells. You can see the structure outside, a cell, another cell, and this is the cleavage furrow. You can see the cleavage furrow here, and this is the nucleus which is getting rearranged. The nuclear membrane is again reforming. You can see here in a plant cell, this is one single cell. You can see the cytoplasmic plate in the between. So it is going to get into two different cells, and this is the two nuclei within the same cell. This is the final stage. Next. Next. Such as pictures of it. We have already seen that. So what is the significance of mitosis? Mitosis is responsible for the development of zygote into an adult. Formation of zygote, that is formation of sperm and egg, will take place by meiosis. But after that, that single cell getting into an embryo, everything takes place by what is called as mitosis only. So equal distribution of chromosome to each daughter cell. Because all of you would have studied a little of cleavage in your plus 2 also. What happens? A single zygote, first cleavage, equal division, 2 cells. Second cleavage, again equal division, 4 cells. 2 to 4, 4 to 8, 8 to 16, 16 to 32, right? This is how the division goes in. So it is always an equal division. So whenever it's an equal division, it is only going to be mitosis. Because meiosis is a reduction division. So any equal division will always be brought down into what is called as mitosis. So it is responsible for the growth and development of the individual. It maintains the constant number of chromosomes in all the body cells of the organism. Mitosis is required for asexual reproduction, vegetative propagation in plants, and also responsible for the repair and regeneration of the tissues. It also helps in maintaining purity of the genome because there is no no interchange of any genetic material from the paternal and the maternal. It is just division and doubling up. And it is responsible for repair and regeneration of old and damaged cells in animals. Example, the gut epithelium and the blood cells. So with this, we come to end of mitosis. Next class, we will see into what is called as meiosis. Thank you, friends.